Welcome to the third video in Entomology 6210. My subject for today is going to be modes of speciation. Now, I'm not sure how many students in the class have already had a course in evolution. If you have had, then perhaps some of this material will be review for you, but I'd like to make sure that everybody at least has the basics on this subject. So let's get going. First, concerning the readings. I posted 3 papers for you to read. And as I was rereading them in preparation for this recording, I guess I decided that perhaps the third paper, the one there in gray, the one about scale insects, maybe I was overreaching a little bit there, because a lot of the techniques they use are going to be subject for another part of this course, where we talk about how we use phylogenies to infer things about evolution. So let's not worry too much about that paper at this point in time. And what I'm going to talk about today is going to be focused on the subjects of the Torelli et al. Trends in Ecology and Evolution paper, and the Smodgy and Button paper on chemosensory speciation. So we all know that the landmark book that Darwin published in 1859 was entitled On the Origin of Species by Natural Selection. And that title has always been a bit of a misnomer, in the sense that, well, let's start out with what Darwin thought species were. A couple of your readings have already referred to this. So for Darwin, working in the mid-19th century, he could observe lots of variation in the things in nature, and these things that they observed were classified and put in a standard classification. And one level of this classification was the species, but the species, unlike what we talked about last time, where the species is distinct in that it has, at least purportedly, an objective definition. Excuse me. Wow. For Darwin, the species was just one step in the imperceptible gradation from individual differences within a taxon. As they became more and more extreme, then things were classified at higher and higher levels, so that a species, individual species, would be distinct from one another. And if those distinctions were large enough, then those species would be classified in different genera. And those genera would be different from one another. And if those differences were large enough, then those genera might be classified into different families. And going the other way, there could be small differences within a species that in the old days would be classified as a variety, or perhaps a subspecies. There's a whole list of categories that people used over the years, and we'll talk about more about those next time when we talk about the classification. But the point is that this was a continuum from individual variation through slight variations, through variations that were significant enough to call things different species, to variations that were so significant that the organisms were classified in different families, different orders, even different kingdoms, right? So for Darwin, the origin of species was distilled down to how did these differences arise, and that was really the focus of that book, the mechanism by which such differences could appear, could evolve. And of course, the contribution here was that the mechanism was proposed to be natural selection. Now, I'm not going to go into natural selection to any great extent in this course. I hope that you're all familiar with the concepts. A little bit of jargon that you'll run across is that the gradual accumulation of differences, or evolution in traits within a lineage, let's use that terminology for now, is often referred to as anagenetic change. And I'll give you the contrast here on the next slide. So changes, evolution within a lineage in traits, size, color, morphology, behavior, anything. That's referred to as anagenetic change. And the mechanism for that are the typical mechanisms we talk about in microevolution. You know, it could be natural selection, it could be genetic drift, it could be the appearance of mutations, et cetera, et cetera. So in Darwin's point of view, when that lineage had changed enough through these anagenetic changes, then it had evolved into a new species, hence the title of his book. But today we look at things a little bit, quite a bit differently, at least in some respects. And we look at the process of speciation, the origin of species, to plagiarize Darwin's title again, is not just how much a species changes through time, but actually the splitting of a lineage, Dicaros' metapopulation lineages, into two or potentially more species, typically referred to as, well, they can be referred to as daughter species when you think of the original species splitting and producing two separate species, so daughter species. Sometimes these two separate species are also referred to as sister species. I guess they would be daughters, wouldn't they? I mean sisters for daughters. So this process of splitting of lineages, in contrast to anagenesis, which is the process of change accumulating within a lineage, the process of splitting lineages is referred to as cladogenesis, with the root word clad referring to branches. So the branching of a lineage. So let's think about for a second, what is it that unifies a lineage? And here we're going to harken back a little bit to the different concepts of what constitutes a species. If you are a proponent or an adherent of the biological species concept, and there are many, then it is the capacity to interbreed, that is for individuals to exchange genes, that is the cohesive force that unites that species. If you want to include the time dimension, as a la George Gaylord Simpson, then ancestor-descendant relationships and the inheritance of characteristics and the genetic underpinning of those characteristics is the cohesive force that unites a species. And one way to look at it, that if you look at your lineage as a kind of a gene pool, then the individuals in that lineage are, here's my computer speak coming in, these can be thought of as instantiations of that single gene pool. So it's the gene pool that unites it, and individuals of a species at any point in time are only particular manifestations or instantiations of that gene pool. So the question is, what if that's what holds a lineage together? What does it mean when a lineage splits? If you're an adherent to the biological species concept, this means that there is some sort of mechanism that interferes with the ability of individuals to interbreed. And collectively, these things are referred to as reproductive isolating mechanisms. Now, I'll give you a bit of caution about the concept of reproductive isolating mechanisms. You will run across that term quite a bit in the literature. The caution is to be careful, because some people would view only intrinsic reproductive isolating mechanisms as falling into that category. By that I mean things that prevent or inhibit interbreeding between individuals arise from the genetic makeup of those individuals, that these differences are intrinsic to them. For example, a difference in a pheromone blend. The pheromones that are produced by an individual are governed, at least in large part, by the genes that they have to synthesize those chemicals. And so if there's a change in pheromone blend because of genetic changes, such that a male moth can't find that female moth, then that would be considered an intrinsic reproductive isolating mechanism. Contrast that with extrinsic reproductive isolating mechanisms, such as, oh, what's a good example? How about brown bears? Are the brown bears of Eurasia a different species from the grizzly bears of North America? Do they interbreed? No. Why not? Because, well, one set are in North America and the other set are in Eurasia. And so simply by virtue of them being physically apart from one another, they cannot interbreed. And so some authors are particularly concerned with intrinsic isolating mechanisms and don't put a lot of importance on these extrinsic isolating mechanisms, because obviously they can break down, individuals can migrate, and things like that. I think I personally fall in the camp where both types of isolating mechanisms are potentially important and can separate species. And so we get into perhaps a case of a trivial distinction if two populations are physically separate. Are they by definition different species? And for some purposes, I would think you could say yes, and other purposes you'd say probably no. And it's going to be the individual cases, the details of the individual cases that are going to probably bring you down on one side or another. So just a word of caution when you see those terms. So when we talk about modes of speciation, basically what we're trying to understand are the means by which new species arise. That is, what are the mechanisms by which a lineage that's held together by ancestor-descendant relationships, and in the case of sexual species, by interbreeding, what are the mechanisms by which such a lineage can be subdivided so that as they proceed through time, they're actually evolving independently of one another? So, modes of speciation. I'm going to couch most of this video in terms of the traditional, or I guess I put classical, no, I put traditional here, somewhere else I put classical, method of organizing potential modes of speciation, which are based upon geography, the physical location of the lineages in space and what happens to them. If that doesn't make sense to you, it will in a second. And in that framework, we can talk about three general classes of speciation modes. The first is allopatric speciation. So remember from your general ecology that two populations are considered to be allopatric if they occupy different geographic regions, that they're physically separated from one another. The other side of that coin would be a sympatric speciation, which would be the origin of species when those populations occupy exactly the same space. So in allopatric speciation, new species arise when they're physically separated. Sympatric species, new species arise even though they're occurring in the same geographic area. And then there's a third general category, which is actually probably a minority of cases, or just special cases, which is called parapatric speciation. Be careful here of pronunciation, because we're going to talk about two different concepts. Parapatric, P-A-R-A, patric speciation, which means the origin of new species when they occur in geographically contiguous but not overlapping distributions, so that their distributions abut one another, but they don't, or only minimally overlap. There's going to be another term that's going to pop up here shortly that sounds the same in conventional American slurring of the English language, peripatric speciation, parapatric speciation, which is spelled P-E-R-I instead of P-A-R-A. As we get into this, I also want to, a lot of this basic categorization of modes of speciation is something that really started to take shape during this period of the modern evolutionary synthesis. So we're talking about the 20s, 30s, and 40s into the 50s. So these ideas were arising, and we have to recognize the context in which they were arising. So even though there were brilliant people working during this period of time, they had no understanding of what the structure of DNA was. Genes and genomes and the function of genes were just being discovered. And although people understood the concept of a gene, even though they didn't know exactly what it was, the expectation was that within a species, because individuals can freely interbreed, there we go with the biological species concept again, because they can freely interbreed and exchange genes leading to the next generation, that interbreeding would lead to homogenization of the gene pool. So in order to understand how new species arise, we need to understand how we can disrupt that gene pool, how differences can arise even though within each generation genes are being exchanged. So this is a bit of a conundrum, right? So I'm going to start with the idea of allopatric speciation, which is, well, first of all, I don't think anyone doubts that allopatric speciation occurs. That is, if well, let's talk about the classical model. So in allopatric speciation, we're going to start with a hypothetical parent species that has a distribution in time and space. Let me grab my pointer here. There we go. So here's our parent species, and just these two dimensions. I'm trying to indicate that this is the geographic distribution that this parent species occupies. So we start with a widespread, geographically widespread parent species, and then something occurs that causes that original range to be split. This something is typically, in cases of allopatric speciation, typically thought of as an extrinsic factor, such as, the glaciation, the expansion of glaciers in North America that separated populations between the east and the west. It could be the origin of rivers. It could be orogeny, the origin of mountains. All sorts of things that can occur that would lead to this original geographic distribution of the parent species being split in two, such that we then end up with two populations of the parent species that are isolated from one another. This would be an extrinsic isolating mechanism in this case, a mountain range. But whatever, the parent species has now been subdivided into two daughter populations. When they begin, when that event, which is called a vicariance event, the splitting of the original range, when that occurs, if the populations are large enough, then one would expect them to be genetically more or less homogeneous. They would have the same genetic background against which future evolution can occur. One question would be, even at that point, now that these are no longer interbreeding, are they two distinct species or just the same species on both sides of our hypothetical mountain range? That seems to be a trivial distinction. But now that these two populations are physically separated, then one would predict that the areas in which the two daughter populations now occur are not going to be precisely the same. The population in which the area in which B occurs would be farther north. It's on, if the prevailing winds are coming from the left here, if that's the west, then it's on the leeward side of the mountains, and so one might predict this to be a drier area, and this to be more moist. It's farther south, perhaps warmer. The point is that the environments in which these two populations occur are at least slightly, if not in a major way, different. And therefore, those two populations under different environmental regimens can be expected to evolve in different ways through time in response to the differences in the environment, both the biotic and abiotic components of that of the environments. So these are going to diverge, and because they're not interbreeding, the genes that appear in population A here are not going to be able to find their way into population B through the process of interbreeding. And so in our classical model, perhaps this divergence proceeds to such an extent that those two populations are no longer capable of interbreeding with one another. And if the barrier that originally rose to separate the population now disappears, and the two daughter populations expand in size such that they overlap one another, then they may find that the descendants in population A can no longer interbreed with the descendants in population B, thus fulfilling the definition of a biological species concept, where we have two populations that can't interbreed. This is kind of the classical story of how allopatric speciation could occur. And as I say, I don't think anyone doubts that this occurs, and it probably has probably is a component of almost all cases of the origin of species. So if these two populations have diverged sufficiently in isolation from one another, then they come back together. They might not be able to overbreed. There may also be cases, however. Oh, I'm sorry, I jumped ahead here a little bit. So this is an example of what people think of as a classical example of geographic allopatric speciation. So we have two very similar species of birds, the black-capped chickadee here on the left and the Carolina chickadee here on the right. And if you look at the distribution of these birds, you see that the black-capped chickadee is characteristically distributed in the more northern parts of the country, spreading across the entire width of the continent from the Atlantic to the Pacific, while the Carolina chickadee is more restricted to generally the southeastern parts of the United States. These two species, and these are clearly two different species, they're very easy to distinguish on the basis of, for example, their song. Although, if you just look at the two individuals here and try to distinguish them on the basis of morphological characters, it is very difficult to separate them, especially when these tiny little birds are flitting through the trees and not giving you a chance to get a good look at them. If you're really interested, the black-capped is whiter back here, and it's whiter, more contrasting here in the wings and on the tail. But the songs are clearly different. There's no problem distinguishing between them. And so these are two good species. Their populations do overlap. You can just compare this to this. You can see that there is a narrow area of overlap in the mountains here, although there's probably some elevational differences here in the Appalachians. But across a band going across, and in fact in not all the way north in Ohio, but a little bit farther north than where we are here in Columbus, there is a hybrid zone where these two populations of chickadee species come into contact. Another example, now this is a classic example of allopatric speciation. Not this particular set of toucanets, but the idea, one of the questions was, why are the tropical regions of the world so rich in species, have so many more species within the same sort of geographical area than you find in the temperate zones? If you remember back to Terry Irwin's estimate of the total number of species of insects, within a given area in a tropical forest, say here in the Amazonian forest, you can find literally hundreds of species of trees, while in a similar area in the state of Ohio, you'll be lucky if you'll find 24 species of trees. So we have many more species in the tropics. And so why do we have this? A lot of explanations have been put forward, but one that I find particularly intriguing and interesting incorporates the idea that although in the Amazon basin, or at least before we started cutting all the forest down, this was a continuous wet tropical forest here, more or less. There were some microclimatic differences along the rivers, and depending upon the soils and stuff, you could get some interesting habitats that were different than the wet tropical forest. But you had vast expanses of contiguous tropical forest over thousands of kilometers here. But the environmental conditions that we see today have not been stable over geological time. That we are living, and perhaps we're leaving, but we have been living in a period of relatively high moisture levels, such that these are humid forests across this area. But at earlier periods of glaciation, then the tropics, in this case of South America, were much drier, and the forests that thrived in the humid, wet areas today had actually contracted their populations into areas of higher moisture. So instead of it being broad contiguous forest here, the forest remnants retreated into little pockets up here, along the leeward side of the Andes, down here, over here. And the idea, I think the natural idea is that as these wet tropical forests retreated into these smaller pockets where the environmental conditions were still favorable, the animals that lived in such forests and depended upon such forests also had their ranges, their geographic ranges, subdivided, constricted, that they retreated into these refugial populations, perhaps here in Colombia and Ecuador, thus isolated into these separate pockets. Then when we entered a new wet period, they differentiated in those pockets. And then when the climate got more moist and the forest could expand, things like these toucanets also expanded their range. And then when they came into secondary contact, secondary meaning that their ranges expanded, they were original contiguous population, subdivided into isolated populations, and then came into contact again secondarily, that they had diverged enough such that they were no longer capable of interbreeding. So here we see a series of species of closely related birds in the genus Cylindria. The map in the background is a fairly old map of the distribution of these different forms, and I've superimposed photographs of the birds because, as you can see here, this is the same species as this. It's just much more evocative and colorful to actually see the birds themselves. But what can we see? We can see differences in the size of this ear patch, this big long patch here in this species. Practically nothing there, at least in the yellow, a yellow stripe on this guy. You can see the color yellow around the eyes here, where these other guys are green. Look at the modeling on the color and modeling on the bill. This species is called macula rostris. You can see it has spots, maculae, on the bill. Black tip, green tip, blue tip. Lots of variation there. But the idea is as this continued expansion and contraction of forest habitats was like a pulsing engine for the development of new species. Contraction into refugial populations, differentiation there, then population expansion, they come together. Then another round of isolation, differentiation, and expansion of populations to new ones. So we had this cycling And every time you can imagine that the number of species doubles. So it wouldn't take long before you go from perhaps a single widespread species into dozens and dozens of species. Multiply that by all the species that might have been there in the original population, and you get one possible explanation for why these tropical forests are relatively rich in species. This is not the only suggestion that's been made to account for this, but I think we can safely say that it's got a piece of the truth, if it's not the entire truth there. So I think I want to deal with these 2 points in opposite order. When these populations that were in isolation now come back into contact, their ranges overlap. If they have differentiated sufficiently that they can no longer interbreed, then no problem. We've got a case of allopatric speciation. But it's quite possible that they can interbreed, at least to some extent. And so under such circumstances, we could predict one of 2 things happening. One is that when these 2 partially isolated populations, genetically isolated populations, come into contact, then by the process of interbreeding and gene exchange, then the population will become homogeneous again. Another possibility, though, is that when they come together, that perhaps the hybrids are not as fit in the environment as either of the 2 parental populations. So you can imagine, one can easily imagine selective forces that would lead to the reinforcement of these genetic differences, such that we see the evolution of, or the strengthening of, reproductive isolating mechanisms. In isolation, they may not have evolved to the point where they can't interbreed at all. But once they get together and there's selective forces against the intermediate, the hybrid individuals, then you can see the possibility that the reproductive isolating mechanisms will be strengthened, such that an individual doesn't, quote unquote, waste their gametes on less fit offspring. Also, one would expect, sometimes people subdivide reproductive isolating mechanisms into post-mating and pre-mating isolating mechanisms, or almost the same thing, pre-zygotic and post-zygotic isolating mechanisms. Examples of post-mating, post-zygotic isolating mechanisms would be things that reduce the fitness of hybrids after the hybrids have formed. So it might be reduced fitness, reduced viability, all sorts of things that lead to a hybrid sterility, for example, lead to a hybrid individual not being as fit as individuals that are more purebred from the 2 original hypothetical populations we've been talking about. So in those cases, one would expect the evolution of pre-zygotic, pre-mating isolating mechanisms, such that the parents do not invest their limited genetic currency in the future in offspring that are not going to be able to be as successful as in reproducing and persisting in the population. So when the 2 populations come back together, we could expect either coalescence of the population or reinforcement of any existing isolating mechanisms that keep them from interbreeding and recoalescing. One of the interesting and problems of all of these modes of speciation is that what we're basically talking about are unique historical events. Events that took place at a certain time in a certain place under specific conditions. And so in any particular case of speciation that one might be looking at, it's impossible to actually determine maybe impossible is too strong a word. It's very, very difficult to determine what exactly happened to the parent populations that caused the daughter lineages to split. Speciation itself, except there are some isolated conditions, but generally speciation itself is not directly observable. We're talking about things that took place tens, hundreds of thousands, millions of years ago, and so knowing how and why it took place at that particular time can be very difficult. There are some specific cases, things like, I'm not going to dwell on this too much, but one of the modes of speciation that is not uncommon in plants is the origin of new species by means of polyploidy. This doesn't occur very often in animals, but it's not uncommon in plants, things like ferns. And the idea is that we have the fusion of male and female gametes from two different species. Now, normally the chromosome complements are going to be different, and that would lead to the inability of a hybrid organism to successfully complete meiosis. But if that occurs, and a subsequent doubling of the entire chromosomal complement in that new hybrid occurs, now there's no problem with having homologous chromosomes pair up during the process of meiosis and have meiosis proceed normally to the production of male and female gametes. So that's a case where we can observe speciation taking place almost right in front of our eyes. And there are a few other special cases like that. But by and large, we're talking about unique historical events that we might be able to hypothesize the mechanisms that were involved, but it's rarely something that is amenable to experimentation to actually determine what happened. So, we talked about the classical method, one classical method of allopatric speciation, in which we have a contiguous population that is split by some external force. Another way for this to occur is to have, as we see here on the left, a parent species that butts up against some sort of barrier. And one of the classic examples of this is that this barrier is the ocean. But somehow a small number of individuals from that parent population is able to surpass this barrier and find its way to a new geographic area that they colonize. So this could be the mainland and an island off the mainland. You're probably already thinking of one of the most famous examples, which of course are the Galapagos Islands, which as volcanic islands have always been separated from the west coast of South America. And they have been colonized by individuals that somehow actually didn't go from west to east, but they went the other way from east to west, colonized by species from South America. And now here, in isolation from the parent species population, they have evolved in response to the environmental conditions that they encountered on these islands. So the idea of these founder events is that a small population, which almost by definition is going to be genetically different from its parent species in terms of the relative frequency of alleles in its gene pool, finds its way across a barrier and is able to evolve in isolation, separate from its parent species, and given sufficient time and sufficient rate of evolution that they could evolve into a distinct population. One of the classic examples of this is thought to be the case of the Hawaiian Drosophila. Now, you are probably familiar with Drosophila melanogaster, Drosophila simulans, some of these species are standard lab animals of genetics. These flies, at least traditionally, the classification of these flies is changing, but they're closely related to the workhorse of the genetics labs, Drosophila melanogaster. But Hawaiian Drosophila are a bit larger, and they're a lot more charismatic, Unfortunately, in my picture here, you can't see this one in the lower right-hand side, but you'll be able to see it in the PDFs of these slides. But you can see that there's patterning on the wings, different series of spots, and they differ in their behaviors and courtship behavior. And somehow these flies found their way to the Hawaiian archipelago, which is the most geographically isolated area of the world, situated there thousands of miles from any mainland in every direction. But they did find their way there. And the amazing thing is that in this group of flies, there are now more than 700 species of these flies that are known, all derived from a single or very small number of founder events. And I don't know how familiar you are with the geology of Hawaii, but Hawaii sits atop a hot spot in the Earth's crust, such that we get the volcanoes coming up. Now, the volcanoes erupt, form the islands, and these hot spots are moving under the surface of the Earth, such that the islands up here to the northwest are the oldest islands, and that's where the hot spot used to be. But it has gradually been shifting toward the southeast, and the big island of Hawaii here is now the youngest and most volcanically active island here. You still get some activity going toward the west. And so old to new, series of islands. Diamond Head here is the remnant of the volcanic crater that used to be there, but now all the activity is focused down here on the big island of Hawaii. The fact that these are smaller reflects the erosion that has taken place, and the islands actually continue off farther here to the northwest. But what this map is trying to illustrate, and people have tried to reconstruct what went on with the Drosophila in Hawaii that led to such a tremendous increase in the number of species. And so this deals with just one subset of the total diversity of Hawaiian Drosophila. In Kauai here we have 12 species, Oahu 29 species, Maui and Molokai 40 species, and down Hawaii here 26. And in trying to reconstruct this, it appears that there have been a series of founder events. We had an initial founder event where the ancestor of all of these species found its way into the archipelago. But then from Kauai, there was an invasion, actually 5 different invasions of Oahu with differentiation of species there. One species from Oahu apparently found its way back to Kauai and differentiated there, now in isolation from its parent population. 10 founder events into the Maui complex, and then 7 back to Oahu, 1 all the way back to Kauai. In one case, Kauai got all the way to Maui, 15 into Hawaii. You see this cycling here from Oahu directly to Hawaii. We have this complex interrelated series of founder events where a small group of individuals from the parent population, let's say from Maui, found its way to the Big Island, differentiated there, and so became new species. So instead of one species here, now we have the original and then a daughter species here. So we get this multiplier effect going back and forth. I think you can see how this could be a very complex web of interrelationships to try to unravel. And it's probably becoming at the same time easier and more complex as we have genetic tools to look at these animals to try to figure out what's happened. So in terms of allopatric species, these are the 2 major sort of canonical descriptions of what happens. We have either vicariance, the splitting of an ancestral population into 2 pieces, or more, could be more, or these founder events where some individuals are able to overcome some physical barrier, the ocean, a broad river, a desert, any of these sorts of barriers that you could imagine. There are a couple of other flavors, if you will, of allopatric speciation. This is the one I warned you about, peripatric speciation. And the idea here is similar in many respects, what we've already discussed, but we have a central, more or less contiguous parent population, more or less geographically contiguous parent populations. But perhaps during some periods of time, when environmental conditions are especially favorable, that parent species increases its geographic distribution, but then contracts under less favorable conditions, and it might leave little isolated populations behind as it contracts. And it's in these isolated pockets that differentiation to the point of reproductive isolation can occur. This is, I suppose, the refugia that I discussed in terms of the toucanets might be an example of this, although looking at it from the opposite viewpoint, that we contract into these isolated populations and then expand again to overlap. Another one, another idea that I want to bring in here because it has particular roots within the entomological community, is a mode of speciation called Stacy-Patrick speciation. And the work on this was spearheaded by this gentleman here in the middle, Michael White, MJD White, who was an Australian scientist. And he was particularly interested in studying these grasshoppers. These are morbine grasshoppers. Their family classification has jumped around a little bit. They're now considered a separate family of grasshoppers. These are wingless, rather sedentary grasshoppers that are characteristic of Australia. And so at the time MJD White was working, there was a lot of interest in the techniques available to look at the chromosomes of organisms. And these grasshoppers are characterized by a number of chromosomal rearrangements, transversions in their chromosomes, such that these chromosomal changes made it difficult for individuals with two different configurations of their chromosomes to interbreed, because meiosis wouldn't take place properly. I mean, you could get one generation, but they would be unable to produce viable gametes in the future. And so White's idea was that within a large, more or less contiguous population, we have these very sedentary individuals within it that are not capable of large-scale migrations. So while we might think of this as geographically contiguous, in fact, we seem to have a whole series of subpopulations with perhaps some, but limited gene flow between them. And then within one of these populations, a chromosomal rearrangement could take place, let's say an inversion, such that individuals that from this population that had that inversion could no longer effectively interbreed with individuals from this population that had the parental style of chromosomes. And so he envisioned that the species differences, these isolating mechanisms, in this case the chromosomal rearrangements, could arise in situ, hence the name Stacy-Patrick speciation, that they're static, they're not moving like the founder populations and before. And that, at least in the case of these grasshoppers, and perhaps more broadly, it was the chromosomal structure that was the driving force in speciation in these populations. As attractive as this might seem, I mean, there are obvious problems with this. If you have a chromosomal inversion occurring within this population, how does it become the dominant form of the chromosomes in this population? How does that single event translate into more widespread feature of the entire subpopulation? I also want to point out that more recent work, at the time MJD White was working, there was very limited ability to look at what was actually going on in the genetics of these organisms beyond the morphology of their chromosomes. More recent work done at the University of Adelaide, which is up here, so this is the southeastern corner of Australia, Tasmania. They've looked at allozyme variation amongst the populations of these grasshoppers, the same ones that MJD White was working on, looking at mitochondrial genes, specifically CL1, looking at a couple of nuclear protein coding genes, and just trying to get more data to assess White's model for how the species arose. And the bottom line, I think, at this point anyway, is that White was correct that some of these chromosomal species do seem to be valid in that they correspond with some of these other genetic markers. But it's far from the complete story. This particular picture here, we'll talk about this in lab, but this is a neighbor joining tree of allozyme variation within these things. And there is not only chromosomal variation, but there's a distinct geographic pattern. Let's see, where can we point to? I think E4 and F2, where they occur down here in this area, that there's a geographic distribution of these things, such that it's possible that an allopatric model of speciation could also account for this variation. Within some of White's chromosomal quote-unquote species, we find some of these geographic subdivisions. The Murray River is coming along here, and so that could be separating populations on the east and west side. And conversely, found that there is some gene flow, particularly in the mitochondrial genome, between White's chromosomal species. And so these chromosomal rearrangements apparently are not fully capable of isolating populations. So, as I said, each of these things is a historically unique event, and probably there's lots of features, lots of influences that are going on. Geographic separation, ecological specialization in areas, chromosomal rearrangements, such that the story is more complex than we generally consider. Looks like I misspelled Viatica there. Okay, so let's move on from allopatric speciation to consider the next two components. I don't want to spend a whole lot of time here on the concept of parapatric speciation. This is the idea that two species can arise when their geographic distributions abut one another, where there's an area where the populations come in contact. But for some reason, even though they might be capable of interbreeding, if you took them and put them in a zoo, in fact, in nature, they don't. And typically, cases of parapatric speciation, people invoke the idea that there is you have a continuum of the environment, but that there is a change in that environment across that continuum, such that in, let's say, the far western portion of the population, one set of genes is ideal for that environment, and in the far eastern side of the population, another set of genes is ideal. If the environment, and if this environmental gradient from east to west if there's not an abrupt change from east to west, but just a very gradual change from east to west, then one would expect that there would be gene exchange across it, such that you would go from gradually one set of genes in the east to one set of genes in the west. But if the change in environment is abrupt, such that there is we usually think of these in terms of clines, that the environment changes very rapidly over a short geographic area. Let's not say rapidly, that implies time. Let's say it changes just over in a narrow geographic range. Then the two quote-unquote ideal genotypes could come close to one another and may interbreed in this area where they come together. But those hybrids are selected against in either of the parent populations, and so we do not see introgression of those genes from one population into another. That effectively those two gene pools are kept separate. This gives rise to the idea of ecological speciation, which is getting a lot of attention these days. That what I have seen is the work is usually conducted in the context of the biological species concept. So they're looking for reproductive isolation between populations or segments of populations. But ecological speciation posits that the reproductive isolating mechanisms, those actual factors that keep these two populations from interbreeding, arise because of natural selection. And this would include also sexual selection. It's a bit controversial, and again, trying to demonstrate that selection has taken place and given rise to the differences between populations such that they can no longer interbreed is something that's difficult to do, and you can demonstrate it only under a restricted set of circumstances. But it's certainly getting a lot of attention for where we see populations of things. We'll talk about timemes when we get to talking about walking sticks later in the course, where it seems to be the case that natural selection, in that case for camouflage, could be leading to the isolation of populations and the origin of new species within them. So let's finish up talking about sympatric speciation, an idea that is widespread, keeps popping up, and has traditionally been one of the most controversial suggestions for how new species arise. Because the basic problem is that what Meyer and people in the 40s sort of were hung up on, that how could you get genetically based isolating mechanisms evolving within a population if the individuals in that population were continually exchanging genes from one generation to the next? How could you get segregation of certain sets of genes in one subset of the population and not in another if interbreeding was leading to homogenization of that gene pool? Well, there are some suggestions that have since come up, but as I say, this has been relatively controversial. 20 years ago or more, it was the population genesis mathematical modeling suggested that sympatric speciation could occur, but the conditions under which it could occur were going to be relatively rare, and so it should be unusual. I think the consensus is shifting on that issue. And to lead into this, let's talk about Rhagoletis pomonella, the apple maggot. So I'm not sure if you're familiar with this insect. It is the poster boy, or girl, as it may be, for cases of the study of sympatric speciation. Rhagoletis pomonella, common name the apple maggot, is a member of the family Tephrididae. Here you see an adult, very colorful, very almost charismatic sorts of creatures. In their life history, what happens is that adult flies find a fruit, let's say an apple here, and here we see a female Rhagoletis ovipositing within the apple. She lays her egg underneath the peel of the apple, and her offspring, the maggots of the apple maggot common name, then bore within the fruit, feeding upon it and fungi in there. When the maggots mature and they're ready to pupariate, they emerge from the apple, drop to the ground, pupariate in the soil, and then eventually emerge as adults. The adults then fly to find a new host, and it's on this host that mating takes place. Now, you're probably familiar with the fact that the apple, the commercial apple that we have so many varieties of these days in the grocery store, apples are not a species that is native to North America, whereas the apple maggot is. The native host of the apple maggot are hawthorns. So these little, practically inedible, tiny fruit. They're rosaceous. It's a whole lots of species of Crataegus, the genus here. Lots of hybridization. Very, very difficult taxonomic problem here. But these are the native hosts of these flies. And here we see a fly on the hawthorn, on the fruit. But it seems that sometime, I'll come back to that. No, I won't. I guess I can't go back to that. In the 19th century, the native Ragulatus that normally would feed on Crataegus shifted to mate, reproduce, and feed upon this imported species that Johnny Appleseed and his like were spreading across the continent to apples. And now, because mating takes place on the fruit, the populations that prefer apples for whatever reason, that's where they find their mates. That's where mating takes place. And they no longer are going to be interbreeding with their parent population that is still going to the hawthorns. That they, in this host specialization, have led to a disruption in the reproductive community. Some reproduce on apple, or some mate on apple, and some mate on the hawthorn. And insofar as finding those hosts in the first place is based upon the genetics, then these are now, perhaps they have completed, perhaps they are in the process of evolving separately, even though they live within the same geographic area. And they're vaginal enough that they are capable of, at least theoretically, encountering one another. It's not unusual to find hawthorn and apple physically close to one another. The flies can fly far enough to actually find them if they so wanted, but they don't, because the cues they're using to find their hosts lead the two subsets of this population to be mating and reproducing on different hosts. Then we get into a question of which comes first, the chicken or the egg, because we find lots of physiological and behavioral characteristics that differ between these two. Some people still refer to them as host races. Others are willing to accept that they have become different species within the observational capacity of a single individual. But did these differences arise before or after their isolation? There's different fitness on different hosts. They respond differently to host-produced chemicals, to the colors of the fruit. They have different phenology, timing of when they are on the wing and interested in mating and reproduction. But the basic idea is that host races could be an engine to be developing new species for subdividing this a previously contiguous, reproductively interbreeding population into subsets that now either have reduced or perhaps no interbreeding whatsoever, because the breeding is mediated by their association with these hosts. I don't think I can go back, but I will try. Now, the previous slide, which you'll see in the PDFs, was just a much more recent article that is pointing out that, well, when the apple maggot shifted from hawthorn to apple, that had an impact on in the case of that paper, on the parasitoids of the apple maggot, such that now there's a new population of hosts that the parasitoids could attack that are found not on the hawthorns but are now on the apple. And so once the parasitoids find their way to that, then they differentiate into new species. So you can have one species leading to a cascade of speciation events in its ecologically associated species, in this case, their hymenopteran parasitoids. So basically, Meyer thought back in the 40s and 50s that the isolation geographically of a population and the fact that when they came back together they were no longer able to reproduce, was this inability to reproduce was caused by a genetic revolution that took place in the daughter populations while in isolation, that so many changes had accumulated within the genome that when they got back together they were no longer able to successfully interbreed. And of course, as I tried to emphasize earlier, this was done in the context long before we had the kind of details now that we have about population genetics and how different populations really are. So it's no longer accepted that reproductive isolation presupposes, it depends upon these dramatic, almost comprehensive changes in the genetic makeup of the daughter populations. That it's conceivable that there could be a small number, perhaps even just a single genetic change that could lead to reproductive isolation. One of the sort of whimsical terms that's arisen to refer to such a change are to call them magic traits, that a small change, perhaps as little as a single gene, could have far-reaching impacts, pleiotropic effects, if you will, that impact mate choice, the ability to mate, the ability to find mates, the ability to successfully reproduce. And so one small change could lead to the sort of reproductive isolation that Meyer presupposed depended upon these massive changes to make things reproductively incompatible. I'm particularly enamored of the paper by Smadja that implicates the chemosenses, that is the ability of any organisms, but particularly insects, to their senses of taste and smell. How changes there could impact the reproductive community. Insects are very dependent upon their chemical senses to find mates, think of pheromones, sex pheromones, to find host plants, to find host organisms. And it's entirely conceivable that changes in their ability to detect and respond appropriately to these important environmental cues, important chemical cues, can change their ability to find mates and to successfully reproduce. In other words, that a change in their ability to respond, to detect or respond to chemicals, can lead to this assortative mating that, taken under some circumstances at least, when taken to its natural conclusion, leads to a division of the population into into separate non-interbreeding lineages, i.e. speciation. And in this sense, excuse me, refer to chemosensory speciation because it's based on their ability to respond to these cues and not on the geography of the situation, but their physical proximity to one another. That individuals can be, the population can be contiguous in that they're vaginal enough that at least physically they could come into contact with one another, but because they breed in separate places, because they're responding to different sets of chemical cues, that they don't, that we have assortative mating even though they're physically in the same place. So I think definitely the move here, it's not to say, as I think I've repeated myself here, that allopatric speciation or some of these other methods do not occur, but they're perhaps not the be-all and end-all that traditionally they have been considered considered to be, that the major modes of speciation may be mediated not by location within an environment, but by some of the cues that species are using in order to find potential mates or potential habitats within which to breed. That's at least where I see the future as leading very strongly. So finally, just a quick summary here. Despite what the title of Darwin's book is, speciation is not the accumulation of changes within a lineage, that's anagenetic change, but cladogenetic change, that is the division of lineages into two or more daughter lineages. Classically, the perspectives on how this occur were based upon geography, giving rise to allopatric models of speciation, where divergence arises when they're physically separated. More theoretically controversial sympatric modes of speciation, where somehow divergence occurs even though the individuals are in close enough physical proximity to be able to mate with one another. And then parapatric speciation, where you have steep environmental selective declines that lead populations to be unable to separate and to be unable to interbreed. I think the core feature in speciation is to ask oneself, what is the cause of the disruption in the lineage? And one can focus that on the ecology of the separate lineages, if you have the ecological species concept, their ability to reproduce, if you're a biological species concept, or any of these subsets. This is why de Queiros's idea of a unitary species concept, where we focus on metapopulation lineages and not upon the details of the mechanisms that lead to this disruption, is important. In the cases of ecological speciation, it is natural selection that is leading to the difference. In chemosensory speciation, it's the evolution of the senses of smell and taste. I guess I really want to emphasize this last line, is that none of these are mutually exclusive, that all of these things can be going on at the same time, feeding on one another. And so it becomes a very difficult proposition to be able to assert with any confidence that any particular example, particular historical example, that this divergence occurred because of this set of factors or that set of factors. It's often a complex interaction between the two. Okay, I'm going to quit for now, and next time what we'll jump into is taxonomy and classification, general principles of those. Thank you. I'll get this posted as soon as I can.